Here's a question that completely changed modern microbiology. If one bacterium is too small to matter, when does a bacterium start mattering? The answer turns out to be when there are enough of them, and bacteria, it seems, know exactly how to figure out when that moment has arrived. The process is called quorum sensing. It's the way bacteria communicate, coordinate, and collectively govern their own populations using small chemical signal molecules. And understanding it, truly understanding it, may be one of the most important scientific breakthroughs of the past 50 years. Today we're going to unpack the science of bacterial communication, trace the career of the scientist most responsible for revealing it, Dr. Bonnie Bassler of Princeton University, and explore how her discoveries are pointing toward an entirely new generation of antibacterial medicine. Let's start at the beginning. Bacteria are unicellular organisms. They don't have nervous systems. They don't have brains. And for most of the history of microbiology, scientists assumed they had no meaningful way of coordinating their behavior. They were viewed as fundamentally solitary creatures. That assumption was wrong. Quorum sensing works like this. As bacteria grow and divide, they continuously produce and secrete small chemical molecules called autoinducers into their surrounding environment. When the concentration of autoinducers crosses a threshold, a quorum, the receptor detects this, and a signaling cascade begins. Gene expression changes, new proteins are produced, new behaviors are activated. Think of it like a stadium doing the wave. Nobody goes first until enough people around them are already moving. The behavior is collective, emergent, and coordinated, without any central authority, producing enough toxins to overwhelm a host immune system, building a biofilm complex enough to resist antibiotics, generating enough bioluminescence to serve as camouflage in a light-scattering ocean environment. These are group projects, and quorum sensing is how the group organizes itself. Bassler enrolled at UC Davis, planning to become a veterinarian. A chance assignment to a bacteriology lab redirected her trajectory entirely. She earned her undergraduate degree in biochemistry, then a doctorate from Johns Hopkins. Bassler's lab went on to characterize this second signaling molecule, autoinducer 2, known as AI2. Structurally, AI2 is notable for incorporating boron, an element abundant in marine environments, as a cofactor. In these communities, bacteria aren't just counting members of their own species. They're receiving a complex blend of chemical signals from their entire neighborhood, a molecular cocktail encoding species composition. Who is here? How many? All of this is being computed, constantly in real time, by the bacterial community as a whole. To understand why this matters so urgently, we need to briefly confront the antibiotic resistance crisis. Drug-resistant infections now kill over 1 million people per year globally. Projections suggest this number could climb to 10 million annually by mid-century. We've been in an evolutionary arms race with bacteria for 80 years. Penicillin introduced in the 1940s, resistance reported shortly after. Methicillin in 1960, MRSA emerged in 1961. We need a new philosophy. She has received essentially every significant prize in biological science, the MacArthur Genius Fellowship in 2002, the Shaw Prize, the Wolf Prize, the Canada Gairdner International Award, the Princess of Asturias Award, the National Medal of Science, the Albany Prize, the UNESCO L'Oreal Award for Women in Science. The bacteria have been talking for billions of years. Thanks to Dr. Bonnie Bassler, we finally know what they're saying, and we're learning to change the conversation. For more information, visit the Bassler Lab website, and for more medical detective stories, go to unbedsidemanners.com.